Let's get right into it. Imagine you bought a book titled The Complete Instruction Manual for Being a Human. You open it up, excited to finally understand why you crave pizza at midnight or why you can never remember where you put your keys. But there's a problem. 98% of the book is written in a language you can't read. It looks like nonsense, gibberish, a cosmic practical joke. The other 2%? That has the recipes for making eyeballs and hair and earlobes. Useful, sure, but it doesn't explain much of anything else. For decades, this was exactly how scientists viewed your DNA. The Human Genome Project finished mapping our genetic code back in 2003, and they found that only about 2% of it actually codes for proteins, the building blocks of you. The other 98% was a vast dark expanse of what they cheerfully and very wrongly labeled junk DNA. They figured it was just millions of years of leftover evolutionary clutter. The biological equivalent of that drawer in your kitchen filled with dead batteries, old rubber bands, and a single mysterious key that fits nothing. But here's the secret, the one that's been slowly unraveling in labs all over the world. That junk drawer? It's not a junk drawer. It's the master control panel for the entire house. And some of those switches are wired to some pretty alarming things. This isn't just old genetic baggage. It's a living record of ancient plagues, evolutionary gambles, and biological dark magic that makes you who you are, for better, and sometimes for much, much worse. Your DNA is a hoarder. It never throws anything away. It's got scraps of code from the dawn of multicellular life, fossilized viruses from plagues that swept the earth before humans were even a thing, and countless broken, disabled genes that are just sitting there collecting dust. For a long time, the prevailing wisdom was, well, it's not doing anything, so it must not be important. This was, to put it mildly, like looking at all the wiring inside your walls and concluding it's just there for decoration because it's not actively making toast. This so-called junk is more accurately called non-coding DNA, and its primary job, the one we've only recently begun to appreciate, is gene regulation. It's the operating system. That 2% of your DNA that makes proteins, that's the software, the apps. But the 98% of non-coding DNA is the immensely complex system that tells each cell which apps to run, when to run them, and for how long. It's the difference between a neuron and a skin cell. They both have the exact same library of protein-coding genes, the same DNA book. But the non-coding day in a neuron is acting like a frantic librarian, screaming, okay, we only read the chapters on neurotransmitters and axon construction. Ignore everything about keratin and sweat glands. For the love of all that is holy, do not start growing hair in here. So where's the danger in that? The danger is that this system is fragile. A tiny error, a single misplaced letter in this non-coding junk, can act like a stuck key on a keyboard. A gene that's supposed to be off gets switched on, or a gene that's supposed to be on gets silenced forever. It's not the gene itself that's the problem. It's the ghost in the machine, the faulty instruction coming from the dark matter of your genome. And when the project manager of your cells starts making bad calls, the entire project can go catastrophically wrong. The real secret isn't what the junk DNA does. It's that our own ignorance of it was the most dangerous thing of all. In the 1940s, a geneticist named Barbara McClintock was staring at corn, not in a gosh, I could really go for some popcorn kind of way, but with the intense focus of someone witnessing the laws of physics spontaneously break. She was looking at the patterns of colors in the kernels, and they weren't behaving. According to the genetics of the Maya, traits were supposed to be stable, passed down in an orderly fashion. But her corn kernels were a chaotic mess of splotches and streaks, changing from generation to generation as if some invisible vandal was sneaking into the corn's DNA and spray painting over the instructions. When she presented her findings, the scientific community gave her a polite, collective pat on the head and basically told her to go sit down. Her ideas were too weird, too radical. What she was proposing was genetic anarchy, and she was right. What Barbara McClintock had discovered were transposable elements, better known by their rock and roll name, jumping genes. These are rogue segments of DNA, often originating from that junk pile, that have the terrifying ability to copy or cut themselves out of one part of the genome and paste themselves somewhere else entirely. Imagine a single sentence in your 3 billion letter instruction manual suddenly getting up and inserting itself into a totally different paragraph. Most of the time, the result is gibberish. If a jumping gene lands in the middle of a crucial gene, say one that helps suppress tumors, it can break it completely. It's a genetic saboteur causing mutations that can lead to diseases like hemophilia or cancer. This isn't just a faulty switch, like in our last example. This is an agent of pure chaos, actively rewriting your code without your permission. But here's the twist. This genomic vandal is also a master artist. Over evolutionary time, this constant shuffling and reshuffling is a primary driver of innovation. A jumping gene might land near a dormant gene and accidentally switch it on. Or two jumping genes might grab the chunk of DNA between them and move it somewhere new, creating a brand new hybrid gene with a totally new function. Our own evolution, the very things that make us human, was accelerated by this internal chaos engine. It's dangerous, absolutely. But it's also the reason we're not still pond scum. Decades after her work was dismissed, Barbara McClintock was awarded the Nobel Prize. The secret wasn't just that our DNA was alive with these jumping vandals, but that the biggest piece of junk in the whole story was the scientific dogma that refused to believe it. What if I told you that you are, biologically speaking, about 8% ancient virus? Not that you have a virus, but that the virus is you. Woven into the very fabric of your DNA, sitting in every single one of your trillions of cells, are the fossilized remains of infections that plagued your distant, non-human ancestors. These are called endogenous retroviruses, or ERVs. Here's how it happens. A special type of virus called a retrovirus infects a host. Its whole game plan is to inject its genetic material into the host cells and merge with their DNA. Now, 99% of the time, this is just a personal problem for that one infected animal. But every once in a million infections, that virus strikes gold. It hits a sperm or an egg cell. And if that specific cell goes on to create an offspring, that viral code is now a permanent part of the family line. It gets passed down from generation to generation, a ghost in the machine. Your genome isn't just a blueprint for a human. It's a graveyard, a permanent archaeological record of the plagues our lineage survived. For a long time, these were considered the absolute peak of junk DNA. They were broken, deactivated viruses, just taking up space. But then we looked closer, and the story got much, much weirder. Our bodies, in an act of stunning evolutionary jujitsu, have harnessed these ancient enemies. We put them to work. The most dramatic example is a protein called syncytin. It's absolutely essential for the formation of the human placenta, the organ that allows a fetus to get nutrients from its mother. Without syncytin, mammalian pregnancy as we know it would be impossible. And syncytin is, you guessed it, a repurposed viral gene. The original gene was used by an ancient virus to fuse cells together so it could spread. Our bodies co-opted that exact function, not for infection, but for creation, fusing cells to build the wall between mother and child. The dangerous secret here is a mind-bender. One of the most fundamental aspects of human life was stolen from the toolkit of a prehistoric plague. The thing that tried to wipe out our ancestors is now the reason you exist at all. Think of your cells as cars. To work properly, a car needs two basic things: an accelerator to make it go, and a brake to make it stop. Simple enough. Cancer, at its core, is a disease of broken cars. It's what happens when a cell's accelerator gets welded to the floor and, at the same exact time, someone cuts the brake lines. The cell starts dividing uncontrollably, and it can't stop. It just goes and goes and goes, creating a pileup of dysfunctional, malignant cells. For years, we hunted for the cause of this by looking at the car's engine, the protein-coding genes themselves. We found genes that act as accelerators, oncogenes, and genes that act as brakes, and tumor suppressors. But that was only half the story. The terrifying truth we've now uncovered is that in a huge number of cancers, the engine is perfectly fine. The genes themselves are not broken. The problem is in the wiring. The problem is in the junk DNA. Those accelerator and brake pedals aren't part of the engine. They are regulatory switches located in the vast, non-coding regions of your genome. A single tiny mutation, a typo in the junk, can permanently jam the go signal for an oncogene, telling it to floor it forever. Another typo in a different stretch of junk can sever the connection to a tumor suppressor, effectively cutting the brakes. The blueprint for the brake pads is still there, but the cell has lost the ability to press the pedal. Scientists have now linked mutations in these non-coding regions to all sorts of cancers, from prostate to brain to chronic lymphocytic leukemia. The dangerous secret here is insidious. It means that the seeds of our most feared disease aren't necessarily lurking in some monstrous, mutated gene. They're hiding in plain sight in the boring bits, the parts we ignored for half a century. It's like discovering the safety manual for a nuclear reactor is full of typos, and realizing that a single misplaced comma could be the difference between normal operation and a full-scale meltdown. You know those little plastic tips at the end of your shoelaces? They're called aglets. Their only job is to keep the shoelace from fraying and unraveling into a useless piece of fluff. Now imagine if every time you tied your shoes, those little plastic tips got a tiny bit shorter. Eventually they'd wear down to nothing, and your shoelace would be toast. Your DNA has the exact same problem, and it's a problem that's slowly killing you. At the end of each one of your chromosomes, you have a long, repetitive cap of non-coding DNA. It's pure junk, in the old sense of the word, just the same sequence of letters, T-A-G-G-G, repeated over and over. These caps are called telomeres, and just like aglets, their job is to protect the important genetic information in the chromosome from getting damaged every time a cell divides. But here's the catch. Every single time a cell divides, those telomeres get shorter. It's a planned obsolescence built into your very being. This shortening acts as a biological clock. When the telomeres get too short after 40, 50, or 60 divisions, the cell hits a crisis point. It can no longer divide safely without risking damage to the actual genes. So it does one of two things. It either triggers a self-destruct sequence, or it enters a zombie-like state called senescence, where it stops dividing but hangs around, causing inflammation and contributing to what we experience as aging. Wrinkles, a weaker immune system, fragile bones. Much of this is the macroscopic result of trillions of microscopic shoelaces getting too short. So where's the danger? Can't we just stop them from shortening? We actually have an enzyme called telomerase that can rebuild them, but it's usually switched off in most adult cells. Why? Because the one type of cell that famously reactivates telomerase to achieve immortality is cancer. By reactivating this enzyme, cancer cells solve their shortening shoelace problem and can divide forever. The secret hiding in this piece of junk DNA is the ultimate biological catch-22. The very mechanism that stops your cells from becoming immortal tumors is the same mechanism that ensures you will grow old and die. Your mortality is the price you pay for cancer prevention, and the receipt is written in your telomeres. You would think, after decades of work and billions of dollars spent mapping the human genome, that we'd at least have the full book, even if we couldn't read it all. You would be wrong. It turns out there are entire sections of our DNA, vast stretches of non-coding regions, that are so bizarrely structured, so maddeningly repetitive, that our best sequencing technology simply chokes on them. They are the blank spots on our genetic map. These regions are collectively known as the Dark Genome. And it's here that some of our most troubling secrets are likely hiding. For a long time, these unmappable regions were written off. The assumption was that if they were that repetitive and convoluted, they couldn't possibly be doing anything important. It was the junk DNA argument all over again, but on a whole new level of ignorance. But as our tools get better, we're starting to get flickering glimpses into this darkness, and what we're seeing is unnerving. These dark regions are not silent. They are incredibly active, particularly in the brain. And when they have errors, they are linked to some of the most complex and least understood human conditions, like schizophrenia, bipolar disorder, and autism spectrum disorder. The danger here is the danger of the complete unknown. We have strong statistical links telling us that something in these dark regions is contributing to these conditions. But we often can't tell what the specific gene or sequence is or what it's doing. It's like trying to fix a software bug in a program where half the code is corrupted and unreadable. You know the bug is in there somewhere, but you can't even open the files to look. The secret of the dark genome is that we thought we were exploring a continent, but we've just discovered there's a whole other continent hidden beneath it, and we have no idea how to get there yet. The scariest part of the haunted house isn't the ghost you can see. It's the noise coming from behind the door you can't open. We like to think of ourselves as unified beings. I want to be healthy. I want to survive. We think of our DNA as a team, all working together for the good of the organism, for me. This is a comforting, sentimental, and completely false view of reality. The truth is, your genome is a battleground. It is a ruthless ecosystem of competing elements, all trying to ensure their own survival and replication, sometimes at the direct expense of the body they inhabit. And this intragenomic conflict is orchestrated almost entirely within the junk DNA. The most visceral example of this is the molecular tug-of-war that happens in the womb, a battle between the genes you inherit from your father and the genes you inherit from your mother. This isn't metaphorical. It's a real physiological conflict called genomic imprinting. Through chemical tags that are placed on the DNA, tags located in the non-coding regions, either the paternal or maternal copy of a gene is silenced. Think about it from the gene's perspective. A father's genes, to put it bluntly, want his offspring to be as big and strong as possible to ensure their own survival, even if it drains the mother of resources. So paternally expressed genes will push for rapid growth and aggressive acquisition of nutrients from the placenta. A mother's genes, however, have a different interest. They want her to survive the pregnancy so she can have more children in the future, spreading more copies of her genes. So maternally expressed genes often act as the brakes, trying to restrict growth and conserve resources. You, developing as a fetus, were the rope in this tug-of-war. For 9 months, a silent, microscopic war was waged inside you, with your father's genetic legacy screaming, "Grow," and your mother screaming, "Whoa, slow down." When this fragile truce works, you get a healthy baby. But if the imprinting fails, if a typo in the junk DNA causes one side to be silenced when it shouldn't be, or vice versa, the results can be catastrophic, leading to severe developmental disorders. The dangerous secret is that you aren't a single entity. You are a compromise, the result of a genetic civil war fought before you were even born. So we've established that the 98% of your DNA we call junk is a minefield. It's a graveyard of ancient plagues, a playground for genetic vandals, a source of cancer, and the ticking clock of your own mortality. It would be very easy to come away thinking that your genome is just a terrible, incompetent design, but that would be missing the most profound secret of all. All that danger, all that chaos, all that junk, it's the reason you're a human and not a chimpanzee. Scientists have identified regions of the genome called human accelerated regions, or HARs. These are stretches of DNA that have remained virtually unchanged for millions of years across all sorts of species, mice, chickens, chimps, but show a burst of rapid, dramatic changes specifically in the human lineage. They are, in a sense, the genetic hotspots of human evolution, and the vast majority of them are not in protein-coding genes. They are in the junk. Take HAR1. It's the most accelerated of them all. It's a piece of DNA that was stable for 300 million years, and then in the few million years separating us from our last common ancestor with chimps, it went completely haywire and mutated like crazy. This gene doesn't code for a protein. It codes for a piece of non-coding RNA that is intensely active in the developing human brain, right at the time when our cortex is forming its complex 6-layer structure. While we can't say it's the gene for human intelligence, it's undeniably one of the key architects, an architect forged in the chaotic fires of the junk pile. Our unique brains, our ability to use language, our capacity for complex thought, all of these revolutionary human traits are tied back to mistakes and rapid changes in the very DNA we once dismissed as useless. The ultimate dangerous secret, then, is a paradox. The junk DNA isn't just a source of internal threats. It is the engine of our creation. Its instability, its messiness, its willingness to break the rules and try new insane combinations is what allowed us to become, well, us. The danger of cancer and genetic disease is the price we pay for the evolutionary plasticity that gave us consciousness. Your own intelligence is a beautiful, improbable accident, built from the spare parts and forgotten garbage of your own genome. And that's our list. If you want to keep feeding your algorithm with more of this delightful nonsense, you know the drill. That subscribe button isn't going to click itself.